Welcome to this video on the TCA cycle or tricarboxylic acid cycle, also called the Krebs cycles. This is the final common metabolic pathway of carbohydrate, fats and proteins. It takes place in the mitochondrial matrix where acetyl-CoA is oxidized to carbon dioxide, generating ATP along with NADH and FADH2, which finally enters the electron transport chain to generate ATP. In this video, we will explore its tissue and subcellular location, detailed mechanism, energetics, regulation, and significance. Let us begin with the introduction to the TCA cycle. This is known by other names also, such as Citric Acid Cycle or Krebs cycles, who discovered this in 1937 and got Nobel Prize in 1953. It occurs in the mitochondrial matrix of all the cells and completes in 8 steps utilizing 8 key enzymes. It begins with the condensation of oxaloacetate with acetyl-CoA, regenerating oxaloacetate at the end of cycle. Nutrients are completely oxidized into water and carbon dioxide, producing 3 NADH and 1 FADH2 as reducing potential and 1 GTP molecules by substrate level phosphorylation. It also provides entry and exit points for various intermediate through anaplerotic reactions, indicating flexibility of the cycle. Let us explore the location of TCA cycle. It occurs in the mitochondrial matrix of every nucleated cells except for succinate dehydrogenase, which is present in the inner mitochondrial membrane. All other remaining enzymes are present in the matrix, including citrate synthase, aconitase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase complex, succinyl-CoA synthetase, fumarase, and malate dehydrogenase. Let us discuss the mechanism of TCA cycle. I will focus on three aspects of this cycle. The first one is identifying enzymes and the steps. Second, generation of the reducing potential NADH and FADH2. And third, identifying the source of carbon atoms which are lost during this cycle. The cycle basically begins with the condensation of oxaloacetate with acetyl-CoA forming citrate, a 6-carbon compound, and tricarboxylic acid by the action of citrate synthase enzyme. This enzyme is a, is a regulatory enzyme. We will discuss in the next slide its regulatory mechanism. Citrate is isomerized to isocitrate by aconitase, which is then converted into alpha-ketoglutarate by the action of isocitrate dehydrogenase. This enzyme performs oxidative decarboxylation, releasing thereby CO2, carbon dioxide, which is the source of this carbon is oxaloacetate, and one or the first NADH molecule is produced in this step here. Alpha-ketoglutarate is converted into succinyl-CoA by alpha-ketoglutarate dehydrogenase. This enzyme also performed oxidative decarboxylation, releasing second carbon as CO2, and the carbon source of carbon is again oxaloacetate, and second NADH molecule is produced. So succinyl-CoA is a 4-carbon compound here, which is converted into succinate by the action of succinyl-CoA synthetase, producing 1 GTP molecule. GTP is equivalent to ATP. Succinate is converted into fumarate by succinate dehydrogenase enzyme. At this step, there is formation of FADH2, reducing potential. Fumarate is converted into malate by the action of fumarase, and malate is converted into oxaloacetate by malate dehydrogenase. So the third NADH molecule is produced during this step, and you can see oxaloacetate is regenerated, and since this is regenerated, this is said to be the catalyst of TCA cycle. Oxaloacetate is derived from pyruvate by the action of pyruvate carboxylase from carbohydrate basically. Therefore it is said in biochemistry that fats are burnt under the wick of carbohydrate. So let us discuss regulation of TCA cycle. There are three key regulatory enzymes. First one is citrate synthase, second isocitrate dehydrogenase, third is alpha ketoglutarate dehydrogenase. All these enzymes are inhibited by the high energy state indicated by high level of citrate, high level of succinyl CoA, or high level of ATP and NADH. All these conditions will inhibit these enzymes and therefore TCA cycle. However, under the low energy state, these enzymes are induced and therefore TCA cycle is activated. So let us discuss the amount of energy produced during TCA cycle. So this is the TCA cycle given here. You can see there is a production of 3 NADH molecule, 1 FADH2 and 1 GTP molecule. NADH and FADH2 after undergoing electron transport chain will produce ATP. One NADH molecule produce equivalent to 3 ATP molecule, and one FADH2 will produce 2 ATP molecule. Therefore you can calculate the amount of ATP produced per cycle. So 3 NADH will give you 9 ATP, one FADH2 will give you 2 ATP, one GTP will give you 1 ATP. So total will be 12 ATP molecule per round of cycle, or per acetyl CoA molecule. TCA cycle is extremely important metabolic pathway in the energy production, particularly in the form of NADH and FADH2, which after undergoing electron transport chain will produce massive amount of ATP serving biological function. In addition, it also provides precursors or substrate for various biosynthetic pathways such as fatty acid, cholesterol, amino acid, heme biosynthesis, and gluconeogenesis. In addition, this is a major integrative pathway with carbohydrate, fats, and protein metabolism, energy metabolism. Clinically, this can also serve as a very important target for drugs, particularly in case of cancer, to starve them of energy and killing cancerous cells. Thank you very much for watching this video. Please like it, share it, and subscribe it. Until next time, you take care. Bye bye.